Hello and welcome to this lesson on the cardiovascular system. So let's jump right in, and as you can see, I've drawn a little diagram of the heart here, and let's just do some labeling right now, right off the bat. Let's do some labeling. So we'll start over here at the superior vena cava, and then the inferior vena cava. Superior meaning above and inferior meaning below. Then we'll go on to the right atrium. And then we have the tricuspid valve. I'm just going to write tricuspid valve. And a trick that I learned in human anatomy in high school is this little phrase called, I'll use this color, try to be right. So I'll write that down. Try to be right. So the tricuspid valve is on the right side of your heart. Okay, now the right ventricle, right here, then into the pulmonary valve. It's this thing right here, but I'm just going to label it as P valve. All right, and then the pulmonary artery. Pulmonary artery. And then the pulmonary veins. That's over here. And over on the other side. Okay, into the left atrium. And okay, so this valve right here, I'm certain that a lot of your books will synonymously call it the bicuspid valve and the mitral valve, but I'll tell you right now, in the real world, no one says bicuspid valve. It's the mitral valve. So I'm just going to say mitral, okay? Then this is the left ventricle. And then this valve right here is the aortic valve. I'm going to say A valve. A valve. It's kind of hard to see. And then finally, into the aorta. This guy up here. Okay, now we can talk about blood flow. So I just ordered everything. I labeled everything in order of blood flow, but we'll go through it just to be clear. So we start at the superior and inferior vena cava, which bring deoxygenated blood in from like your head up here and from your abdomen and legs down here into the right atrium and through the tricuspid valve to the right ventricle. And I'll just pause for a second, and I know looking at this, it looks like the right atrium and right ventricles are actually on the left, but you have to keep in mind that right now we have a ventral view of the heart, ventral just meaning like looking at someone's chest. So you would have to take this image and kind of flip it around to like paste it in your own chest, in which case then the right atrium and right ventricles would be on the right side of your body. Okay, from the right ventricle, we flow in through the pulmonary valve into the pulmonary artery, going towards the lungs. Now we'll pause right here. Artery. An artery is carrying deoxygenated blood. I'm sure that most of you, when you think of an artery, you think, okay, oxygenated blood, right? Well, this is the case in the systemic cardiovascular system, which is just between your heart and your body. But in the pulmonary system, it's going between your heart and your lungs. So a way that I always remembered the distinction between what is an artery and what is a vein is arteries go away from the heart and veins come into the heart. So I'll just write that down. Artery versus vein. So artery goes away and vein comes in. I hope that is clear there. So back to what we were doing here. We'll use red for oxygenated now. So then from the lungs, we come back in through the pulmonary veins here, going into the left atrium, through the mitral valve, into the left ventricle, through the aortic valve, out through the aorta, and to the rest of the body in these directions. And you don't have to worry about the names of these guys. I'm sure you don't. It's really just these things that I've labeled here. So now I think, well, I hope it's somewhat clear in this drawing, but I'll go over it just in case. So I think the main defining feature between the right and left ventricle is that the right ventricle has a smaller, I want to say, cardiac muscle density, versus the left ventricle is much thicker over here. Reason being, the left ventricle has to propel blood from your heart to everywhere in your body, versus the right ventricle just has to go to your lungs. So obviously you're going to need more muscle to get all the way wherever else you need to go, Okay. A little bit more about the heart pumping. So let's just go over systole and diastole. So systole is, you know what? I'll write it out here. Systole, spelled systole as such. This is ventricular contraction. So ventricular contraction. So, and during this time, the AV valves, AV just meaning aortic ventricular, the valves between the aorta and the ventricles are closed. Okay, so the reason being because when the ventricles are pumping blood through their respective arteries, they're trying to go this way. So you really don't want blood to go backwards into the atrium. Therefore, it would make sense that these guys are closed at that time. Then diastole, during diastole, the heart is relaxed. Heart is relaxed, and the semilunar valves are closed. So what the heck is a semilunar valve, right? So it's the pulmonary aortic valves. So the reason they're called semilunar, let me see if I can draw it out. So if you're looking directly at those valves, so they kind of just look like our Mercedes-Benz sign from above or below. That's what they look like. If you look at them from the side, though, wow, terrible. From the side, they kind of look like this. There's three parts to them, and they kind of make like a crescent shape, hence semilunar. I'm gonna write that semilunar there, if you can read my chicken scratch. All right, one last thing before we move on. We're going to talk about cardiac output real quick. So I'm sure we've all seen the equation CO equals HR times SV. So cardiac output equals heart rate times stroke volume. So what that really means, cardiac output is measuring the volume of blood being pumped by the heart, both through the left and right ventricle, per unit time. So how much blood is leaving the heart per unit time? That's all that is. That's cardiac output. And you solve that by doing heart rate times stroke volume. Okay, now we're going to go on to conduction of the heart. So the electrical system, I suppose you could say. Okay, and again, let's just get some labeling out of the way. Start with the SA node is right here. SA just meaning sinoatrial node. I'll write that out. So sinoatrial node, which is also, I'm going to say it's the pacemaker of the heart. And we'll come back to that in a second. So then we have the AV node right here. AV node. And AV just means atrioventricular. Atrioventricular, just like the AV valves, there's an AV node. Then we have the bundle of His right here. Bundle of His is capitalized, don't really know why, but it is. And then we have both of these guys, move down. These guys are the bundle branches. Alright, bundle branches. And then lastly, we have the Purkinje fibers. So that's like here and like here, and on the other side as well over here. But I'll just stick to labeling on one side because that's already kind of crowded. So Purkinje fibers. All right. Now the pathway of this electrical signaling. So the sinoatrial node, again, like I said, is the pacemaker. So it literally sets your heart rate. That's why it's called the pacemaker. So electrical conduction in the heart starts at the SA node and goes to the AV node. And I'll just draw out some arrows of how that signal kind of looks. So it goes like, and then also across to here to the other atria. Okay. From the SA node, the signal goes down this way through the bundle of His, okay, and down, it splits off between the bundle branches, right, like that. And from the bundle branches, you go up through the Purkinje fibers, this way and the other way. And that's it. That's it. That's the electrical system of the heart. I'm pretty sure that's the majority of the information you'll need to know on that. I don't think there's anything else serious that you'll have to know. Okay. All right, vasculature. So this is talking about the arteries, veins, and capillaries. So we'll do some labeling. We'll start with just labeling what is what. So artery, this is the artery. This is an artery. And then on the opposite side is a vein. And what's in the middle over here, right? So we have arterioles. Okay, and then capillaries. And then venules. So, Blood flow goes, from an artery through arterioles, which is smaller versions of arteries, then through capillaries, then back through the venules, which is a smaller version of veins, and then through the veins to go wherever else. Alright. Now we'll label the different parts of what is in what makes up these different vasculature systems. So I didn't really leave a lot of room to write. I'll write hopefully small. That's the endothelium, is the innermost lining of a vasculature system. Then you have the smooth muscle. Smooth muscle. Okay. Then we have the connective tissue. That lines the outside of our vasculature. So, all right. Let's talk about arteries first. So, arteries, they're thick and muscular. So the muscle density in an artery is much greater. So I'm going to say muscle over here is increased. They're also fairly elastic, which allows a certain amount of recoil that helps propel blood, wherever it needs to go. Then we'll move on to capillaries. In capillaries, they are 1 cell thick, which means that red blood cells have to travel single file through them. And the capillaries are the site of gas and solute exchange. I just realized that I forgot to label the capillaries. So this is again the and then we have the the basement membrane. It has its own name. So, oof. Right, real small there. Okay. So, yeah, capillaries are super tiny. So red blood cells, have to travel single file through these, which actually, is optimal because it allows the most amount of surface to have that gas and solute exchange. Surface area, that's the term I was looking for. Then moving on to veins. The veins are then a little bit different. So, actually, where's the right color? Here we go. Veins are thinner than arteries, and they're actually inelastic, but they're able to stretch more to accommodate larger volumes of blood. But because they're inelastic, they don't have any kind of recoil. So what really helps blood move through the veins is the compression by the surrounding skeletal muscle. And additionally, I don't know if you can really see, but veins have one-way valves that help maintain flow. So just like I suppose they look closest to the semilunar valves in the heart. So they have like valves right here that then when blood is traveling this way, the valves close when pressure is pushed back on them, and it keeps the deoxygenated blood from flowing all the way back where it came from. And so, what do I want to say about this? I want to say valves. And just clarify that. Okay, yeah. That's that on vasculature. Ooh, blood, 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 blood. Okay. So with blood, well, I have this little test tube over here, you know, so I'm just gonna label that test tube real quick. Test tube, it's really a tube of centrifuged blood, but whatever. So this yellowish part here is plasma, so, which is actually about 90% water, and then like 10% like salts and proteins. So we'll do okay. This yellowish, no, I'm sorry, white. This white part here, key that it's white because it contains white blood cells and platelets. So let's say WBCs and platelets. And finally, the red portion down here, as you can probably guess, is your red blood cells. This is also called hematocrit. So I'll write that out. Hematocrit. That's specifically, it measures the percentage of blood composed of erythrocytes, or red blood cells, and I'll get into that in a second. Alright, so first up here I have some white blood cells that I will label for you. So I divided them into granular and agranular, but oh, I should mention that, okay, white blood cells, their fancy formal name in science is leukocytes. So I should write that down. Up here somewhere. Leukocytes. Okay. So the granular ones up here, these ones, you can see that they have little dots in them. That's the granules, what makes them granular. We have a neutrophil, then a eosinophil, it's a weird name, but, and then a basophil. Cool. So that's the granulocytes, and I don't think you'll have to know how to distinguish between, neutrophil, eosinophil, and basophil, but you should have a general idea that these are the granulocytes. That's, yeah. Then agranulocytes, I have a lymphocyte here, so, and this big guy is a monocyte. Okay, then before I move on, I want to make one final note. So we know that white blood cells, sorry, those are platelets, white blood cells play a really big part in our immune system, right? So a note that I'd like to make, even though this isn't necessarily, this is more, has to do with the immune system than it does the cardiovascular system, but anyways. Granulocytes, these guys up here, play a large role in the innate immune system. So innate just meaning that it is, we're born with this kind of immune system, and that's a lesson for another time. Then lymphocyte and monocyte, the agranulocytes they play a huge role in the adaptive immune system, which is not, that's something that we develop over time. It's what we use vaccines for. That helps build our adaptive immune system. Okay, moving on. Platelets. The fancy name for a platelet is a thrombocyte. I'll write that out. Yeah, no. C. Okay, and then platelet. So you have an idea. Okay, so platelets are, I'll get into blood clot formation later on, but platelets themselves are just cell fragments of megakaryocytes. That's the most important thing I can say right now. So lastly, over here, we have I have a drawing of a red blood cell here, right here, and the fancy name for a red blood cell is erythrocyte. So I will also write that out. Ooh. And that's red blood cell. Okay. So, do red blood cells have organelles? They do not. They have no organelles. They're almost blank inside except for hemoglobin, which I will get into. So, organelles? No, big fat no. They do not. And instead, we have, what color should I use? I want to use red, because that's what makes red blood cells red. Hemoglobin. M-O. Or as I will, it's hardly legible. As I will be referring to hemoglobin from now on is I shorten it to HB. There, now you can read it. So, hemoglobin. What is hemoglobin? It's a protein that carries oxygen. It is the protein that carries oxygen. So, I'll put in parentheses. Okay. So each hemoglobin has 4 heme groups that all bind to one O2 molecule. So I'll write that out. And then 4 heme groups each. And 1 O2. So there's like, gosh, there's hundreds of millions of hemoglobin proteins per each red blood cell. So that's a lot of oxygen that each red blood cell can carry. So I'm also going to write that out. Hundreds. So much oxygen. Last thing I want to talk about in the blood category is blood types. Let's do this in green. So blood types. I'm sure most of you are familiar with the 3 different quote-unquote blood well, no, there's 4 different blood types, but there's 2 different kinds of proteins, and then there's one that doesn't have I'll just, you know what, I'll draw it out. So we use A, B, and O to describe the different blood types. A and B are codominant. Whereas O is recessive. So O. And then, so I said 4 blood types because you can have A, you can have B, you can have O, and because A and B are codominant, you can have AB blood type, which is, I think, really the rarest kind, at least in the U.S. It's the least common. And finally, the last thing that defines your blood type is the Rh factor. So that's the positive and negative part of your blood type. So Rh, come on, positive, negative. So Rh just meaning rhesus monkey, because this positive and negative factor was first discovered or first studied in the rhesus monkey. Oh, one last note I should say. So for A, B, and O, we are born with antibodies for any kind of A and B alleles that we don't have. So, for example, if you're a B blood type, you have antibodies for the A. And if you're O, you have antibodies for both A and B, right? For the Rh factor, the positive and negative, if you're a negative type, then you're not born with anti-Rh antibodies until you've been exposed to it. So if you're a negative blood type and you've never had exposure to a positive Rh factor, you're not going to have that kind of antibody. Okay. All right. Moving on. Blood pressure and solute exchange. So, let's get some definitions out of the way, shall we? So, blood pressure is maintained by baroreceptors and chemoreceptors. So, baroreceptors, I'm gonna label baroreceptors as stretch. Put it probably not in yellow. And then chemoreceptors will also do green, is, I'm gonna say chemical. But I'll get into that in a second. So, we'll do orange. So baroreceptors are actually, they're mechanoreceptors. So they're mechanoreceptors located in the carotid sinus and aortic arch. Mechanoreceptors, that sense pressure changes via change in tension of the arterial wall. So I'll write that down too. There's that. Okay, chemoreceptors. So, overall, they sense a change in pH, but we all know that pH can be affected by a number of things, but chemoreceptors, so I'll say sense So mostly they sense change in pH via carbon dioxide and oxygen, you know, the most common molecules that you'll see in the blood. So for an example, I'll do acidification due to CO2 Stimulation during exercise. Yeah, so it would sense the chemoreceptors in your blood vessels. They would sense this increased amount of carbon dioxide. Let's see. What else did I want to say? So, oh, yes, blood pressure. Blood pressure in itself. We'll talk about the systolic versus diastolic BP. So, systolic BP. I'll write it right here. Systolic BP is, it's measured, I remember I talked about systole and diastole, so systolic blood pressure is measured during systole. So, I'll just write systole. And then, so that is like, it has increased pressure on the arterial walls, so increased. Okay, then there's diastolic BP, which I'm sure you now can guess is measured during diastole. So, and that has, well, okay, decreased pressure on arterial walls, so decreased. Okay, that's that on that. So, low blood pressure. When we have low blood pressure, there are a couple things that we can do to raise that blood pressure. The ones that I'm going to talk about right now are aldosterone and, antidiuretic hormone, also known as ADH or vasopressin. So I like to say ADH because it's the fastest thing to say. So aldosterone works by increasing the amount of sodium retained in the body, and therefore it draws water back into the body. So because water will always follow solutes, so I'll just write increased. Oh, I meant to say increased, so therefore increased H2O. So then ADH has a slightly different way of doing it. So also increases water retention, but via aquaporins in the and I know it's definitely in the collecting duct, but I think also in the DCT of a nephron. So increased via aquaporins. Right on. So last thing, so solute exchange. So I'm just gonna do some quick definitions of hydrostatic pressure versus osmotic pressure. And I'll write this in purple. So hydrostatic pressure is the pressure of the fluid within the blood vessel. Hence hydrostatic. So I will write out pressure of the fluid within okay. Then osmotic pressure is, well, it's the sucking pressure that draws water towards solutes. So instead of like hydrostatic pressure is kind of, you can think of it as like pushing, osmotic pressure is like pulling. So if you have more solutes on one side of a wall, and there's not as many solutes on this other side of the wall, you're gonna, instead of trying to push the solutes this way, it is much easier to withdraw water that way towards the area with much more solutes to try and balance out that solute water ratio. So that's what I mean by sucking pressure. So I'll write that down. Right on. So, yes. Okay. Oh, love this. Love this. Okay. So, that was 100% sarcasm. I used to hate the oxyhemoglobin dissociation curve. I always learned it as like HbO2, which, is synonymous to oxyhemoglobin, but it's easier to write out HbO2, so that's what I do. So the HbO2 dissociation curve was the hardest thing for me to learn for the longest time. But I did some work and I get it now. So we'll just go through a few things here. So we'll start with cooperative binding. What is cooperative binding? Well, hemoglobin is what exhibits this cooperative binding, which is just, it means that each successive oxygen bound to a hemoglobin increases the affinity of other hemoglobin subunits to bind oxygen, if that makes sense. And then vice versa, each successive oxygen released from hemoglobin then decreases the affinity of other subunits. So I have an actual graph here later, but the best way I can describe that is each time an oxygen is bound to hemoglobin, the more and more oxygens that bind to hemoglobin, the more and more hemoglobin that is going to want to bind to oxygen. But as oxygen is released from hemoglobin, like when you get to the tissues and you release oxygen into the tissues, then it's going to become, I guess, easier and easier for hemoglobin to release that oxygen, as it doesn't want to hold on to it as much. Anyways, I'll write down the definition of cooperative binding here. So, HB, again, meaning hemoglobin. I'll say, and vice versa. Alright, now we'll talk about some things that affect this affinity for oxygen. So, so I have this stuff written out here that I'm just gonna use as an example real quick. So, let's say we have an increased partial pressure of carbon dioxide in the blood. So that's what that's it. The P meaning partial pressure of CO2 in the blood. So let's say that is increased. Then that's gonna increase the amount of hydrogen ions we have in the blood. That's also gonna increase, which of course then decreases pH. We get more acidic the more H plus we have in the blood. And so I will explain that via this end-all be-all equation here that I'm sure we all know and love. So to explain this, so you have carbon dioxide and water. You start out with that in the blood, and then in the red blood cell, you use, well, I guess I'll write this out as I go. You use an enzyme called carbonic anhydrase. So carbonic anhydrase to change this CO2 and H2O into H2CO3, and this occurs in the red blood cell. So once you have this, then it just, you don't need it. So you need carbonic anhydrase in order to go from here to here, but once you're here, you don't need any other kind of enzyme or anything to help you go from here to here. So, alright, what did I want to say? Oh yes, okay. So carbonic anhydrase turns CO2 and H2O into carbonic acid. That's, use a different color. So carbonic acid, we'll say. Carbonic acid. Then carbonic acid can freely change to the bicarbonate ion. That's what that is. And hydrogen ion. So, to come back to this, why does the partial pressure of carbon dioxide have anything to do with how much hydrogen is in the blood? Well, it all has to do with this equation right here. So you have carbon dioxide. If this goes up, then we go all the way down through this equation, and it ends up being that this goes up, because carbon dioxide isn't really soluble in the blood. So carbon dioxide is largely carried in the blood in the form of carbonic acid, or bicarbonate or hydrogen. So carbon dioxide, because it's nonpolar and, like I said, not particularly soluble, while bicarbonate and hydrogen ions and carbonic acid are quite polar and highly soluble. So essentially that's the whole reason for having carbonic anhydrase, is so that you can transport carbon dioxide in a much easier fashion. So if you have an increased amount of carbon dioxide, then because we don't like to carry carbon dioxide itself through the blood, we try to transfer it this way. So if you have more of this, you're going to have more of this. And this and this, but related to this equation up here is I'll use yellow. So that comes down like that. So that's how that works. All right, now we'll talk about, actually before I move on, something else that decreases the acidity. Decreases the pH, increases the acidity of the blood is, I'll write it out. I'll write out the equation. So it's when you're doing anaerobic respiration that you end up building up lactic acid. So let's write that out. NADH. It's NADH and pyruvate that goes between NAD plus, and I didn't really leave enough room here, and lactic acid. All right, so yeah, this also decreases the pH of the blood, increases acidity, yada yada. So I just wanted to mention that. Now, we will move on to what makes a left or right shift in the oxyhemoglobin dissociation curve. So this also took a long time to learn, but we'll use yellow again. So if you have a left shift in the curve, we'll start there. No, you know what? We'll start with a right shift. So a high partial pressure of carbon dioxide, which also, as we saw up here, results in an elevated amount of hydrogen ions in the blood, which then results in a decreased pH, as well as then higher temperature and increased concentration of 2,3 BPG, BPG meaning bisphosphoglycerate. I suppose I'll write that out. So let's see, bisphospho. Where should I write it? Bisphosphoglycerate. Right, 2,3. 2,3 bisphosphoglycerate. All right. So that causes a right shift in the oxyhemoglobin dissociation curve. So what does that mean? What that means is, let's see here, you have a decrease in hemoglobin affinity for oxygen. So that, to translate, means more oxygen to the tissues. Right? So a way I liked to remember this is you will have a right shift in the oxyhemoglobin dissociation curve if you're exercising. Exercising is a one of the main things that will do this. So I just remembered that exercise is the right thing to do. You know? So highlight that actually. Exercise is right. Okay. So then left shift is, well, the opposite. So you have decrease, which means decrease, which means increase, and also lower temp and lower 2,3 bisphosphoglycerate causes a left shift, and so that's an increase affinity for, and again, the opposite of what we wrote over here means less. O2 to tissues. All right. Okay. I think that's all I wanted to cover on this page. Here we have my example of the curve. Dun, dun, dun. So here is here's what I guess you would see. So hemoglobin, I have, you know, this hemoglobin, and it has these 4 heme groups, right? So when the partial pressure of oxygen, partial pressure of oxygen to, the partial pressure of oxygen to PO2 is low, as well as when the oxygen saturation is low, your hemoglobin is, there's no oxygen bound to it. I wrote out a little key here, but as you increase, you go this way, you know, you start to, these heme groups start to bind oxygen more and more and more. So as I wrote here, affinity increases as oxygen binds. So remember what I said about cooperative binding, that once one starts binding, the rest start to bind as well, until you have a fully bound or fully oxygenated hemoglobin. So, and then what would look like a left or right shift. So left shift would be something like that. That's probably what it would look like in comparison to the norm here, and then a right shift would probably look like that. So they all end up at this level, but it just, there's some space between here and space between here. Okay. Alright, coagulation cascade. Phew! Let's do some labeling. So here we have a damaged blood vessel, right? So, oops. So an injury to the vessel lining then triggers the release of clotting factors. So, oh my gosh. So clotting factors. And then clotting factors stimulate a change from, it causes or initiates prothrombin. So prothrombin is over here. Oh, I thought something was wrong. Prothrombin transforms into thrombin. Okay? Then thrombin causes a change of fibrinogen. So fibrinogen spelling. Fibrinogen, which is soluble, then fibrinogen turns into fibrin, which is insoluble. Oh, so we form, from the damaged blood vessel, we form a platelet plug. So these little guys here are my platelets. So they make a plug by, well, making a clot, forming a clot of red blood cells to close off that hole. And then through this cascade, we end up with fibrin, right? And fibrin it's these little strands here. It's maybe a little bit hard to see, but it's fibrin. Alright. And then this is officially the blood clot. You know, that's the clot. So the fibrin strands adhere to this plug to form a clot. Alright, and a note, just one note I wanted to make about clotting factors, something that I learned. So clotting factors are, I'll just say right here, synthesized in, so clotting factors are synthesized in the liver. And then something else is that clots, I'll write down here. Clot is broken down by plasmin. All right. Dang, I think that's it. All right. So I hope this was informative for you. If you have any questions, feel free to DM me. And yeah, thanks for joining.